everybody. Welcome to the second lecture on the heart. So we're going to talk about heart valves today. Got four heart valves. They are the two atrioventricular and the two semilunar. All your heart valves are going to work to prevent backflow. Basically, the atrioventricular are going to prevent backflow between the ventricles and the atria. We got two of those guys, tricuspid on the right, we see it right here, and bicuspid or mitral on the left, we see it right there. Two semilunar valves are going to prevent backflow from the great vessels, i.e. the pulmonary trunk and aorta, back to the ventricles. We got the pulmonary semilunar preventing backflow from the trunk here to the right ventricle, and the aorta preventing backflow from the aorta to the left ventricle. All right, let's talk about what makes these valves open. So here's the deal. Valves are going to open when blood is going the right way. So in the case of the tricuspid valve, the right way is from the atrium, the right atrium, to the right ventricle. That's only going to occur, and thus the valve is only going to be open when right atrial pressure exceeds right ventricular pressure. Similarly, the mitral valve is going to open when blood is moving from the left atrium to left ventricle. That's only going to occur when left atrial pressure exceeds left ventricular pressure. All right. What about closing them? Well, it's kind of like the opposite. These valves, these AV valves, are going to close when blood tries to go the wrong way. So the tricuspid valve is going to close when blood tries to go from the right ventricle back up to the right atrium. That's going to happen when right atrial pressure is less than right ventricular pressure. Similarly, the mitral valve is going to be closed when left atrial pressure is less than left ventricular pressure. All right, one more important thing about these valves. Remember, we talked about this in lab too. These valves have a swinging door type hinge. As such, there is the potential for them to flip upward. All right, we prevent that by anchoring the valve flaps with strings of collagen called chordae tenonnae, and then anchoring those strings to the papillary muscles in the ventricles. And what that's going to do is prevent the valve flaps from swinging upward. And that swinging upward is known as prolapse. All right. Let's talk about the semilunar valves for a second. They're different from the AV valves in that they have no danger of prolapsing. They don't have a swinging door type hinge. They're pocket valves, or pouch valves. I like the kangaroo pouch analogy. And as such, they don't need chordae tenonnae. They don't need papillary muscles. All right, let's talk about opening and closing these guys. The pulmonary semilunar valve is going to be open when blood is going the correct way, from the ventricle to the trunk. That's going to happen when ventricular pressure exceeds trunk pressure. Likewise, the aortic valve is going to open when blood's moving the right way, from the left ventricle to the aorta. And that's going to occur when left ventricle pressure exceeds aortic pressure. You've probably figured out the closing of these guys. It's pretty straightforward. They're going to close when blood's trying to go the wrong way. The wrong way is from the vessel back into the ventricle. Pulmonary semilunar valve is going to close when the ventricle pressure is less than that of the pulmonary trunk. Aortic is going to close when left ventricle pressure is less than the aortic pressure. Because in those situations, blood's going to try and go backwards, and those valves are going to get closed. All right, moving right along here. Got a couple of series of questions here. We're going to cover these in class, but you should do them before you get there. So we got a bunch here. I'll skip over all of them right now. We'll talk about them in class. And now we're looking at the myocardium. It is the middle layer of the heart, the muscular layer in between the epi and endocardium. It is mostly made, not surprisingly, based on its name, it is mostly made of cardiac muscle. But that's not all. That is not all. It also contains a connective tissue structure called the fibrous skeleton of the heart. Okay? Let's talk about that a little bit too. We're talking about both these things. The fibrous skeleton of the heart is this dense connective tissue, forms rings and plates around and between the valve openings. And what it does is several things, okay? It provides a place for the cardiac muscle cells themselves in the myocardium to grab onto. In other words, it's their origin and insertion. It also physically and electrically separates the atria from the ventricles. And that electrical separation is going to come back and be really important later on, as we shall see. All right, onward to the muscle, the cardiac muscle in the myocardium. About 99% of your cardiac muscle cells in the myocardium are known as contractile cells. They contract and generate the pumping force of the heart. As we look at them here, we can see the typical striations, the pink, light pink, dark pink lines here that are vertical in this pic. And that, of course, is the result of the overlap of the contractile proteins, which you know already are actin and myosin. We also see some branching going on here. That's a characteristic of these cells. They're not just long, filamentous ones like skeletal muscle was. See some nice nuclei in here as well. And most importantly, perhaps, is we see these reddish vertical structures. These are the intercalated discs. And these intercalated discs are going to link cardiac muscle cells together in two important ways. Okay, in the intercalated discs, we have these passageways made of proteins that span the cytoplasm of one muscle cell and the cytoplasm of another. And what this does is it allows ions to flow from cell to cell. And that electrically joins the cells, allows them to synchronize their depolarization, and allows them to synchronize their contraction, which is the only way they're going to be able to generate enough force to move blood. Meanwhile, we have these proteinaceous zippers that are locking cells together, mechanically, physically joining them, preventing them from tearing away from one another during a powerful contraction. All right, so that's in our intercalated discs. Okay, autorhythmic cells. That's the other 1% of our cardiac muscle. And I'm actually going to pick up there next time. So I will see you guys later.